My question is, did any of you try doing these last two exercises using the unknown sequence, or were you able to get the unknown sequence from the Google Drive? Assets Google Drive. I uploaded an Excel sheet there, and from there you can get the unknown sequence. There I think 50 amino acid residues in each sequence. In each lane there is one sequence. So just copy paste one sequence, and I think I mentioned tons of them, maybe 60, 70 sequences. So just pick any sequence, the entire row, and then do the exercises and see if you learn something, you're getting the same results or you're getting different results. Or you can also practice these exercises using the sequence that I used, that I mentioned in the very first exercise and also in the second one. I don't know. I'm not too sure if I mentioned in this exercise also, today's exercise. Okay, so I think we should start. Today's class is very interested in the sense that we will be also learning programming today. And the way I designed that exercise is just should be good for a person who has no experience in computer languages. They should be still able to write down the commands and try to interpret the meaning of what they are writing and what they are the results they are getting. So today's exercise is about, multiple sequence alignment and phylogenetic tree of an unknown protein. The unknown protein is the sequence that I gave you. Before I start talking about the exercise, this is a journal announcement that I always mention in my last two classes. I also talked about it. This is the package that I'm teaching you from. This package has the instructions how to do the exercises, the videos for all the exercises, plus the some assessment options. So even if you want to incorporate this package into your own course, you can do so. You are most welcome. You need to just download the package on your computer and incorporate the LMS version into your LMS. LMS means Learning Management System. That is the here in North America, all the universities, they use it, where they upload all the lectures, the lecture related materials, and also administer all their exams through those sites. So if you want to incorporate this package into your LMS, I uploaded on this website, the website that's mentioned in here, the package for LMS. You just download it and then incorporate into your LMS. Then you'll be having everything incorporated in your LMS without much effort. So this is a little bit announcement about the asset. If this is the first class you're joining in, you missed last two classes. For those people, the asset, I'm presenting this workshop under the umbrella of ASSET, which stands for American Society of Science, Engineering, and Technology, an international registered scientific organization, and it has a bunch of activities and resources available for young scientists from the school, elementary and high school, as well as undergrads, grads, PhDs, postdocs, and even for faculties. They have some grants available. They have different courses and workshops that they offer from time to time. They are having a conference in the second week of December this year. I would highly encourage you to please go to this link and look around and then register for that conference. It's a good opportunity for people who rarely find opportunities to be able to present. It's a hybrid conference. Hybrid means consists of both virtual and in person. The ones who cannot attend personally, in person, they can attend virtually. In person, it is in Boston. And much more detail you can find if you go to that link. So what we have done so far, we have done two exercises in last 2 weeks. In last week, we did two exercises. Today is a third exercise. And what we have done in those exercises, I have tried to summarize it here. In the first exercise, we used these databases from the unknown amino acid sequence. So you had got the amino acid sequence, and then you're looking for the identity of that amino acid sequence, which protein it belongs to. And then from you try to accumulate as much information as you can about that amino acid sequence. In the second exercise, this one here, we searched all these databases I mentioned here, or some of them, and we learn different aspects about the oligonucleotides and how oligonucleotides also can be used to identify the protein that they specify. Today's exercise, actually, we'll be doing two exercises today. The first one is the multiple sequence alignment and phylogenetic analysis. And second is the R studio and R programming. So I'll teach you what R programming is very briefly, not in too much detail. And I will show you some of the commands that you can write in the R studio to get the information about your protein of interest. The objective of the first exercise are mentioned here, and the question that we're trying to answer or trying to address is, what is the possible evolutionary relationship between a set of sequences and your protein sequence, and what is the predicted protein structure? This is the information we're trying to get. Though we already have got a bunch of information from through exercises, but in case you're relying only on MSA, multiple sequence alignment, or phylogenetic analysis, then you can also find out all the information about your protein using only these two tools. So this is the database that we'll be using, UniProt, and we have used this database in last two exercises also. It's mainly having information about the proteins, their description, functions, domain structure, and modifications, as well as mutations in the proteins, which have been reported by scientists from time to time over the years, are all combined together under the umbrella of this database. What is multiple sequence alignment? Multiple sequence alignment, when you have an unknown amino acid sequence and you're trying to align that sequence with the existing amino acid sequences which are found in the database. In order to do MSA, you need to have at least 3 sequences, at least 3. More, you could have as many as you want, but at least you should have 3 sequences to compare that sequence with the available sequences which are found in the database that I'll show you to search. Right? MSA tools. What happens when you copy those 3 sequences at least and paste them in the database search box, then those databases, they have built-in tools available. One of the built-in tools that we're going to use is called Cholesterol Omega program. And this program or this tool helps with the finding the sequences similar to your sequence of the 3 sequences that you'll paste in the box, right? And as we understand, these tools, they have a built-in algorithm which is designed for protein alignments. So when you, rather than going to using a programming language, understanding and learning how to use it, and then writing the commands, and then do the MSA, here what you have to do is simply just copy the sequences that you want to make an alignment for, or paste them in the search box and click hit or submit, and the built-in tool will do all the jobs for you, right? Now, what is phylogenetic analysis? Phylogenetic analysis, it tries to find you the evolutionary partners of your protein of interest, right? So at the time of evolution, when the proteins were evolving from one single origin and they were then differentiated into different proteins, through phylogenetic analysis, you learn who are the evolutionary partners of your protein. And the reason we try to have this information, in case if you come across a protein, which is a novel protein, and you have no idea about its structure or function, then through phylogenetic analysis, you can find similar proteins in the databases. And then from there, you can predict the structure as well as the function of that protein. So it will help, it usually helps to reach to the, an applied stage as quickly as possible. Suppose if you find out, for example, take the example of this COVID, the viral proteins which were found in the blood of the people who were having COVID. And the scientists, they isolate the viral genome or the viral peptide from the blood of the people. However, they didn't know the identity of, for example, I'm not a COVID scientist, but I'm just asking you, assume that they didn't know the identity of that protein, but they were in a pressure to find a treatment as quickly as possible. Why? Because thousands of people were dying every day, right? So in that case, what they do, this quickly go to these databases, and then try to match the sequence of that viral protein with the available sequences. And from there, they try to make the conclusion to try to find out the structure of the protein as soon as possible, the function of the viral protein as soon as possible, and to look for if there are some ligands available already in the database, or they are approved as drug and already being marketed that we can use to get rid of that viral, particles from our blood, right? So this is how these researches help you to reach to the conclusion and get to the applied results as quickly as possible. Now, there are different tools available for phylogenetic analysis that are built into these websites. A couple of them I mentioned, and I will be using it. One is called neighbor joining, and another one is called UPGMA, Upjuma or Upjuma. And so these tools, you use them at different locations. In neighboring journal, you use when you have a smaller number of entries, when you have two or three sequences at least, and then you're trying to find the evolutionary relationship between those sequences, or you want the database through this tool to let you know what are the evolutionary partners. So you have a smaller number of sequences, then go for neighbor joining. If you have huge number of sequences they're trying to match and looking for the phylogenetic origin of that protein, then you click on the UPGMA option. Then we'll also learn about one software called MEGA, MEGA. I'll talk about this in more detail in one of the coming slides. That software you will have to download, and it doesn't take much room on the computer. It's very easy to download, and it's very easy to use. So once you have the software in your computer, you don't need to go to the databases. You can just simply open the software and then write down the sequence of your protein, do the analysis, and you will get the results immediately, right? So this MEGA is a useful software in constructing the phylogenesis, visualizing those, and also for data conversion. So it's not a very simple tool for only visualization of letting you know these are the partners of your protein. It can do much more than what you think that you can use it for. It can also easily convert alignment files to other formats. Sometimes what happens, we are using different softwares and different tools. They are built in for different type of file formats. And for that, we always look for softwares or apps or tools that can help us to convert the file into the proper format. And I will show you when we do the docking study in the last exercise that sometimes you need to change the format of the information available that you download from the database, because that format is the one which is available on the database originally is not compatible with your software. So you have to change the format of that file so that your software should be able to read the file. The MEGA can also help with that function if you want to. Okay, now start with the exercise. The very first step is we are using UniProt database, the one I mentioned in the intro. This is the database we also have used in the last couple exercises. Here is the link. Copy paste it into your browser. A page like this will show up, and here you will paste the original amino acid sequence, the one that you take from the Excel sheet, right? To paste the sequence, the original one from the Excel sheet, the one that we use in the first exercise and the second exercise. I'm using the one that I use in last two exercises, and I know this sequence is from a protein called urease, which is an enzyme and produced by a bacteria, Helicobacter pylori. And I got all this information by doing the last two exercises. So once you copy paste your sequence, hit blast, you see a run, you just click run blast. It will take some time depending on the speed of your computer. For me, it took only a couple seconds. Sometimes for me also it takes more than a minute. And once it is done, then it will show something like this, completed. Click on completed. When you click on completed, a web page like this will open. That web page has information about all the proteins which have a sequence similar to your sequence that you pasted into the search box on the last slide, right? On the UniProt database. So here, you'll also get much more information. You see all these tabs: overview, taxonomy, hit distribution, text output, input parameters, API request. So if you click on all these tabs, you will get a different set of information about these proteins. So I would encourage you not as a part of this exercise, but just click on overview, you will see this. Click on taxonomy and see what what more information you are getting. Similarly, click on these tabs one by one and look at what information you're getting. And also here, what is shown here? Okay, it's the name of the entry. They also have an entry name. What is the difference between the two? Click on it and then see why they have entry name and entry differently. What is the protein name? Write down the name of that protein. As I told you that my sequence is similar to urease enzyme and urea, because urease enzyme is consisting of two subunits. My urease enzyme, the one sequence I have, it is closer to urea, right? And then you see all top hits, they are all talking about urease alpha, urea, right? Then organism, in which organism they find, if you see all the top hits, they're showing Helicobacter pylori, right? What is the length of the amino acid residues? All of them, they're showing 238, 238, 238, right? But what is the difference between the two? Why they are categorized under different entries? I will only know once I click the entry, right? So just click the entries and then try to learn as much information as you can about your protein of interest. Here you can, here is also showing you that how the tool uses searches. So it is actually searching the reviewed Swiss prot, the information about your sequence in Swiss prot. This is a different database. If you click here, you will find there's one entry. What is that entry? If you're curious, click it and then see what that entry is about. What is this entry and why is it different? And here on the bottom, if you see, there's a 3D structure. So if you want to know the 3D structure of your protein, of interest, click and then it will take you to the 3D structure of your protein. So for my students, usually I have these questions, and the reason I have these questions, then I make sure they went to the website, they search for the answers, and when they submit their lab report or their worksheet, I want them to write the answers to these questions, and they are supposed to do it with their own sequence. They are not allowed to use my sequence, because in this way, they sometimes students, they just copy paste my answers without going to the website. So what I'd ask them, use your sequence and then find answer to these questions. Here you also see that shortcut menu. And then, here it is telling you the identity, the score, and the e-value. So if you click in any of the hit, it will let you know that what is the identity of that protein with your sequence of interest, what is the bit score, and what is the e-value. And if you remember, I mentioned in the last two exercises, I think the first one, then e-value is better to have e-value as small as possible, because e-value, if is higher, it shows the background noise between your protein and the hit that you get from the searches. The higher the background noise, it means the lower the identity of the two proteins. Bit score has to be highest. It tells you how much your protein is similar to the ones which are reported here. And identity is percent identity. Out of 100 amino acids, if 54 are similar in your sequence of interest and the one which is shown on the web page, it's been this 54% identity, right? Now the next step, what you do, okay, we have not started MSA yet, multiple sequence alignment, and for that you need at least 3 sequences. Remember I mentioned in the beginning. And how would you get those 3 sequences? What was shown on the last page, right? So here you see these boxes, these check boxes. Click on at least 5 check boxes. 1, 2, 3, 4, 5. Don't do more than 5, otherwise you will get very complicated results. And make sure that you do not hit more than 5, right? So I check those 5 hits. This means I want to compare the sequence of these 5 proteins. And once I click these ones, while you do this for the next step, and as I mentioned before, there is no harm in looking at all these parameters and then look what all these tabs, and then look at all the information and features that you can find more about your protein of interest, right? So in the overview, this is what you see, right? Click on these 5 sequences, then click on align, right? So what does it mean? You are asking the web page that I want to do the multiple sequence alignment of these 5 sequences, right? When you do that and you click align, this is what you see. in the align page, the next page. And that page contains 1, 2, 3, 4, 5 sequences, right, of all the proteins that you hit, as well as that's all it contains. It contains the 5 sequences of all those proteins that you hit, and then comparing with each other. So what you have to do, copy all these sequences, all 5 FASTA formatted sequences, because these sequences are shown in the FASTA format. If you look at what I mentioned, if you see that descriptor in the beginning on the left side, it simply shows our FASTA sequence. So when you hit the line, you will get a FASTA sequence of all 5 proteins, copy them, and then here you copy them, you have copied it. That's one thing I'll show you where you paste that copy. But once you have that got sequence, then write and then click on align 5 sequences. So what we are asking the tool that to please align all 5 sequences, the ones that showed up on this page. Once you click that, you will see this one here. A box like this showed up. What is it doing actually? It is aligning all 5 sequences from each protein, right? And it's telling you how much conserved all amino acid residues are in all those 5 sequences. And there is a way to read those sequences. I'll talk about this on the next slide, as soon as you have understood that. And if you're doing that exercise, then I ask the student to write down answer to this question. Write down the position number of first white amino acid in the first row of each of the sequences. So if you see here, there is the all other sequences are bluish, right? But this amino acid residue, which is shown here, is white in all 5 sequences. Here only this one is white. Here only this one is white, right? And what does it mean actually? So how would you read it? There is a way of reading it, but before we do it, either you can read on the last slide, but again, I want you to now come to the cholesterol omega page, and for this, copy this link and paste in your new browser tab. Don't close the old one. Open a new tab and copy paste this sequence. And the 5 fastest sequences that you copied from the previous page, paste those sequences in that page over here, right? And then hit submit. Once you hit submit, you will again get the same information that you get on the previous page, right? So there are 2 ways of finding out that information. And again, many tabs will show up when you have this search box displayed on your computer. Alignments, result summary, guide tree, phylogenetic tree. So even with this cholesterol omega, you can get phylogenetic tree. You don't need to go and find a different tool for phylogenesis. With cholesterol omega, you can also get phylogenetic tree of these proteins, right? So through cholesterol omega, you can get that information. And this information almost similar to what you get on the last page. How to read these sequences? You will see these sequences, like the way they are displayed, there are 5 sequences, but what happened, the way computer displayed it, it broken down the sequences here and then showing the rest of the sequences in the second row, right? And then broken down here and then showing the rest of the sequences in the third row. So there are not 15 sequences, there are 5 sequences shown in different 3 rows, because it's not possible to show all the amino acid sequence in just one row, because there are so many. There are more than 200 amino acid residues, 238 amino acid residues, if you remember what I mentioned before. And if they show it, they will become very small. If you look at the bottom page, line, the bottom row in these sequences, and unfortunately, I cannot show it because I'm not on the web page and I cannot zoom it. But if you zoom in or zoom out, maybe I think you can do it. I have no idea if Zoom has that function. So if you zoom in, and then you'd be able to see this one more properly. And I've zoomed in already for you here. If you look at this red box, and here you look at these stars. So these stars are asterisk. If you see asterisk, it shows a single fully conserved residue. This means all the amino acid residues that you're seeing here with asterisks at the bottom, they're all fully conserved. They're all found in all 5 proteins, right? If you see a colon, this is a colon that you're seeing in here, it shows conservation between amino acid groups of similar proteins. It shows that if this K, which is an amino acid residue, single letter representation of amino acid residue, which is for lysine, if I believe. So this lysine is conserved in similar proteins. So if the 5 proteins are similar, then you will see this colon sign. A period dash like this, it shows conservation between amino acid groups of weakly similar proteins. So the colon is telling you that the conservation is between the similar proteins. The proteins are similar in the sense they are very much related in structure and function. And period means the amino acid sequence is similar in proteins which are not so much related within in the structure and function. And this is a question that I ask my students, that what is the position of the first non-conserved residue? If you see a space, then it shows that that amino acid residue is not conserved, right? And this is something the students have to find out. Now, with the cholesterol omega, as I mentioned before, you can also look at the phylogenetic tree of all those 5 proteins. So this is the a tab that you click on the same page. We were in alignment tab before. Now, if you click on the phylogenetic tree tab, you will see 5 proteins here and how they are related to each other. From this, the tree that I'm seeing here, this tree is also called cladogram or a phylogenetic tree or it's also called phylogeny. I think I'll show you on the next slide when I talk about phylogenetic tool. So here what you're seeing, how you can interpret it, that the first 2 proteins, they are very closely related. The first 2 proteins. And these 2 proteins are related closely to the third protein, but not as much as they are related to each other. And then these 3 proteins, they are related to last 2 proteins, but not as much as they are related with each other. That's how you read the phylogenetic tree. Now, if you have posted more than 5 sequences, if you have posted the sequence of more than 5 proteins, you will see a tree like this. In that case, when you're asking cholesterol omega to do phylogenetic analysis for you, you will click on neighbor joining method. There is a check mark that you have to click on to find out which one you want to use. And because there are more than 3 sequences or the number of sequences, you have to check neighbor joining method. The tool will use neighbor joining method to analyze your protein sequence. If they are less than less than 5, but more than 3, then click on the checkbox UPGM, right? And if you have more than 5 proteins, then this is something that you see. And how to read it again? So it means this protein that you're seeing here, it's more related to these 2 proteins. This one. If you look at here, there's more related to these 2 proteins. And this protein is very much closely related to this one. This protein, the identity of this protein with this protein is more than the identity of this protein to this protein in the bottom, right? So this is how you learn about, if suppose my sequence is this one and I'm looking for a similar sequence, I will pick this one. For this protein, the most closest sequence out of all the sequence displayed here is this one, the one which is displayed next to it. And this is my amino acid sequence or nucleotide sequence, and I don't have any information about it. I just discovered it in the blood of the patient, and I want to know what that protein is about. And I find this one is phylogenetically more related. I'll go and it's been information about this sequence is available in the database, right? I'll go and pick it up and then learn about this protein, that what it does, what's the function, what's the structure, right? This is the cholesterol omega, and mega is software that I talked about. Mega software is very easy to use, and I want you to download that software. If you have Windows, from this link, and if you have Mac, from this link. Once you download it, the instructions to use it are very easy. They have been mentioned in this YouTube video. It's only, I think, 3 to 5 minutes long video. I don't exactly remember, 3 or 5 minutes. Very easy. Like they have mentioned very simple steps how you should use Mega. So if you go to Mega, download this software, and then click on this link to get the YouTube instructions, how to use this software. And then once you find out how to use it, which should not take more than 5 to 10 minutes of your time. This is what I tell my students. And then I ask them, what is the difference you found in doing MSA using Cluster Omega or Mega program? This should be MEGA, not MDGA. It's a typo over here, right? So that's all about the phylogenetic analysis and multiple sequence alignment. And we'll move on to the R program. But before we move on to R program, I have a question for you. Why do plants hate math? Any idea? You can write your answers in the chat. I see somebody already wrote something in the chat. Sorry, because I cannot see chat while I'm presenting unless I click on it, and then only I can see. So you write your answers in the question. What do you think? Why do plants hate math? They don't like math. Many students I know, they don't like math. If they are you, especially if you're a biology student from physiology background or cell biology background, they don't like math. But even plants don't like math. Okay, Muzammil asked a question that my first lecture, can you please sequence to be used for searching in BLAST? I think I showed in the beginning, Muzammil, that how can you BLAST. The very first step where you copy paste your sequence, go to the site on the UniProt, the link I provided, and then copy paste your sequence and then hit BLAST. So simple, it's not difficult. And I think once this workshop is done, you'll be having access to all the videos. And then from there also, if you go through the video, you can have a look and then see how did I mention the steps. But they're not very different than the one I mentioned today. So I'm just looking for an answer from students that why do plants hate math? Any idea? It's a fun riddle. Then I want you to write down. There is no wrong answer. You can try to see if you know the right answer. You can write in the chat. So, okay, I think the people are having a hard time to guess. Why do plants hate math? And I said it's a fun riddle. And the reason is because it gives them square roots. Square roots is a mathematical term and also related to the plants. That's why, it's just we use it as a fun riddle to find out if a student can relate to together. Okay, now the next exercise is protein analysis using R and Bio 3D. What is R and what is Bio 3D? I'll talk about in a little bit more detail. So what we actually will learn through this R programming, we'll learn how to fetch the PDB file of your protein into the R and perform attribute sequence and blast search on that PDB using the information from the PDB file and specify the threshold. And the question that we're trying to address using R, how can we fetch or analyze a protein's attributes from its PDB file? So, the way as I mentioned before, I designed this exercise for that it should be good for the beginners and also the advanced students who actually have some idea about the how to use R. They should also should not find it too easy or, too simple to interpret. They should be also able to learn something out of it. They will able to learn that, okay, writing command is important, but then how to make use of those writing commands, like how to design a project or an exercise based on this R programming language. What is R? R is a programming language that normally scientists use in research in the lab when they work for statistical computing and graphics. So when they're trying to do some statistical analysis between different groups of data, they use R programming. However, we are using it for a little bit of different purpose because this is this is not the learning outcome. Statistical computing is not the learning outcome of this workshop. The workshop is for bioinformatic exercises. So we're using R for doing a bioinformatic exercise. So what is R? Now, in order to use R, you need to use, in fact, two systems. One is called R Studio, another one is R. R Studio is an integrated development environment program for R. right? So this is a program actually that runs all the commands. R runs all the commands, but in order to write those commands, you need RStudio. So it provides a graphical interface. So you don't need to turn your computer into Linux, right? Or any other command language friendly, format. So with your Windows, you can still use this R programming and write the commands and then get the information that you're looking for with the help of RStudio. So when you install R, you are also supposed to install RStudio. And these R studios, they have packages available. For example, if some of you have used Canva, for example, and some other software and tools that we use for making presentations, they sometimes have the built-in templates available. The Canva has, if you're working on infographics, for example, they have built-in infographics template available. You just need to use any of the templates, change the headings or the text or the images, and there you go. Your infographics is ready. Similarly, these programming languages, they have certain packages available. Those packages contain the commands in it. You just need to modify the commands a little bit and take use of those packages. So these are the templates. You can call them as templates, which are built in these programming languages. So we will be using a package is called Bio3D, and this is Bio3D is available in a software repository called CRAN. I also have one more exercise which is not part of this workshop. It's called BioStrings. It's another package, and that package is available from the Bioconductor repository. Now, what you have to do for R programming, you have to install RStudio and R both in your computer. So there are two different sites that you have to go and then both of them and install both of them, and it doesn't take much time and it doesn't take too much space in your computer, right? So you install R using the link. If you click on this link, it will take you to the site. In case the link doesn't work, copy paste this link into your browser, and then from there, if you have Windows, then you click on this install. If you're installing it the first time in your computer, then click on the link here and you'll be able to download the R into your computer, right? So click on this link, go there, and then it will ask you to install R for the first time. Click on that, and then you will find something like this. Previously I was using 4.2.2 version, but when I looked at the website this year when I was offering this course, they have updated it to now 4.3.2 version is available. Install it in your computer. This is the first thing. Second thing, install RStudio. For RStudio, again, go to this link and then download RStudio desktop, this information here, and it will be installed in your computer. You want to know what is R programming, what it can be used for, how to work with it, how to install these two versions that I just mentioned. Then there is a link to about where you can get all the information about R. It's a very good set of information, step-by-step instructions, and written in a very easy, understandable language, so that you should be, even for beginners, it's very, like they should not have any problem once they read the instructions, that how R works and how they can use R. One thing before we move on, that all the commands that I'm writing and I'm showing you through this exercise, they are case sensitive. If a capital letter is mentioned, you have to use a capital letter. If a small letter is mentioned, use a small letter. Not only this, they are space sensitive also. If there is no space mentioned, don't insert any space in your command line. If there is a space, yes, you have to mention the space, right? If there is a colon, you have to make sure that you're writing a colon. If there is an inverted comma, you have to make sure that you're writing the inverted commas. If you're making these mistakes and not very careful in typing or copy pasting the commands, you will end up not having, the command will not work. Now, once you have installed both RStudio and R, go to your start menu and write R there. It will show the R where it is installed in your computer. Click on it and open the RStudio actually. When you open the RStudio software, names written on the pans, but this is what we call them. This console, the one on the top is environment and one on the bottom is plots. The plot, why we call it plot, because if you want to visualize any of the graphics, the plots and tables, they will all show up here on the bottom right, right? Usually you can write commands in console in this panel. How new when you click on file, a pop-up will appear. Click on new file and then R script. When you click on that, one more pan will open in your in this window, and then that is called text editor, right? Or you can, this is a shortcut way of going and opening the R script. If you click on this the plus sign with the green circle, right? Okay, what is the difference? In text editor, whatever the commands you write, they are saved. So suppose if you're troubleshooting, things are not working, you don't have to write everything from the beginning. If you have the commands written there, they will be saved and you can save them. Every time you open the R, you will be able to see the commands. In console, the commands are not saved, right? Now, as long as the file is open, the window is open, the commands should be displayed, but once you close it, you won't be able to see the commands. So for beginners, I say usually that write your commands in text editor and they will show up in console. They also show up sometimes here in the console, in the text editor as well. Okay, though I think this everything that I just showed, which I have written in here, I showed already, talked about it. The first thing you have to do once you open that panel, all these pans, you have to call the library, right? And how to call the library, I'll show you in the first slide. But before, let me talk about this run and source. These keys that you see, that these keys that are usually visible on in the console over here. Run means, suppose if your cursor is, this is a command line, library bio 3D, and your cursor is here. When you click on the, when you press enter here, it will run only this command. The system will run only this command. If you press console rather, then it will run all the commands. That's the only difference between run and console, right? So in case you want to write all the commands first and you don't want to run them, then write them first and then press source, this one here, and all the commands will be run. But if you want to write one command and then want to run it and see how does it work, then you can either press enter or you can press run, right? So these are some, the housekeeping things that you should know when you are start working with this RStudio R. So you open the RStudio. Now you have to call the library. And for calling the library, you go to tools menu. Tools menu just showed up usually here in this place, where you see file edit and code. Go to tools menu and then click on install package. When you click on that, a pop-up will appear like this. And it shows repository CRAN. I think it shows already. And you write Bio 3D. When you're writing Bio 3D, the computer picks up and then the system picks up and then it will start showing you the option. Either you can click the ones that is showing up or you just write Bio 3D and then click install. So once you click install, now you have called the library. It is in your RStudio. Now you can use that package for doing your exercise. And if you want to know what a package does, type question mark 2 times package dash name to bring the associated help file. So you will know everything which is contained in the Bio3D package that you just install in your R. So once you've done that, these are the set of commands that I have written in here, and they are in bold. All the bold stuff that you're seeing, for example, library Bio3D, PDB underscore ID, this arrow sign, a dash, then inverted commas, 1E9Z in inverted commas. So everything that you see bold, these are the command lines. Either you can type those command lines in your console, or you just simply copy paste it from the slides if you have them available, right? And then click enter and you'll be able to see what I'm showing on the right side. So first of all, you call the library, you write library Bio 3D as I have written in here, right? press enter. When you press enter, it will come to the cursor will come to the next line. On the next line, write PDB underscore ID, arrow dash, all this stuff, and there are no spaces in between. And then click on enter. Right? When you click on enter, so what are you doing actually? 1E9Z is for my protein. This is the code. This is a PDB code for my protein. You'll be writing the PDB code of your protein. Right? You can try 1E9Z in the first time when you're doing the exercise, when you're learning like that how the system works. You can try that. But in order to make sure that you actually learn it, try the same all the steps with your own PDB code. The 1E9Z is a code for my protein, which is given to the by the PDB protein database. This one in here, that contains all the information about my protein, this sequence, mutations, its structure, and function, right? So what I'm asking Bio3D package to please install and fetch that information from the PDB site about my protein. This is a site. If you don't know what is the PDB code for your protein, you can go to this site, write down the name of your protein, and from there you can choose that which PDB code that you find closely matching with your protein of interest. Next, you write the PDB and PDB, and then again the same command that I have on E here, and hit enter. All right. Next, type print PDB and hit enter. All right. So when you're writing these commands and you're hitting enter, you're getting information like this after every command. Right. So for example, when you write PDB, read PDB.PDB ID, which I have written in here, this is, and you hit enter, this is the information will show up about your protein of interest, which is fetched by the R Studio from the PDB file that we installed from PDB database 1E9Z, right? It has like total number of atoms, protein atoms, nucleic acid, protein sequence, and and if you scroll down, you'll get even much more information. Sometimes I find if you look at this command line number sign, read and online RCSB, protein data bank, structure PDB, arrow sign dash read dot PDB, and in bracket PDB ID. This I've written in here. Because some students, they complain to me, they say that this command line doesn't work for them. So in that case, I say okay, don't worry, don't use that command line. But you can try. It maybe for you, perhaps it will work. So just write down this command right after PDB ID. When you're done writing this PDB ID 1E9Z, then write this command, this one in here, and press enter. And if it doesn't show any error sign, it means that your system is reading the command. And then when you write PDB, what I just mentioned, this one the code I wrote, the command line, and click enter, this information you get. Then you click print PDB, and the results for print PDB showed in the console, which I'll be showing you in the next slide. The tip is try to use the built-in R functions as they pop up when you type in like read, print, PDB. So when you're typing these code in command lines in your computer, the console will start showing the pop-ups. The way when we write messages in the cell phone or in the computer, if you have any editing language or Grammarly software installed in your computer, then it starts suggesting you the options that, oh, are you writing this? And then you click on it, and then you get that the entire word is displayed on your screen, right? Usually I see it when you're writing messages in the cell phone. Exactly the same thing I'm telling you. When you're writing something and it shows you that pop-up shows up, and you find that the command is the same as you want to type in, click on that. Because in this way, you're trying to sync in with the available, with the R Studio. And then when it sync in, then it will accept your command even faster, and then it will give you the response even faster, right? So this is a suggestion, it's a tip only. You don't have to do it. You think that you're okay by typing yourself, you can still do it, and it should still work, because I chose very simple command lines. So in the console, when you write the attributes PDB like this, then something like this will show up. And attributes mean that you're learning more attributes about your protein or sequence, which are already contained in the PDB file. And all of these attributes, for example, atom, XYZ, remark, what are these attributes? Like what is the information contained under this command line? Write PDB $ATOM and then click enter. You will see something like this showed up. So it will tell you the information about all the atoms, not amino acids, atoms contained in your protein sequence from PDB. Right? Similarly, if you, for example, click on PDB delta remark, REMARK, which I've shown in here, then it tells me that my protein contains two chains, chain alpha, chain beta, right? The similar, you can write this out and find out how many chains are there in your protein of interest. Right? And you can just find all the information just writing the same thing, PDB $XYZ, PDB $SEQRES, PDB $HELIX, and so on. Now, when you write PDB seq in brackets PDB, here you'll get all the amino acid sequence with the numbers associated with each amino acid residue in your console. Right? So it is called, this function will fetch the sequence from the PDB object in the form of an array of characters. If you print the sequence, it would be submitted something like this. So, your sequence all showed up in here. You just copy and paste it wherever you need this information. So what I find that 1E9Z sequence for chain A has 569 amino acid residues. So my question is, for your protein of interest, how many amino acids your protein is composed of? So if you get this information and find, for example, if you look at here, it says 569, right? So this is for my protein of interest. Similarly, for your protein of interest, you can find out how many total amino acid residues it has. Now you wanna see what is the ratio of the amino acid your protein contains? For example, how many alanine residues, how many cysteine, how many aspartic acid, and so on. And for that, write down this sequence, this command line, sequence, arrow sign, dash, PDB seq, and this one, in bracket PDB. Please, it took my half an hour of my time. When I was writing this command, it was not working for me. And I didn't know what was the mistake I was making. The mistake I was making, instead of using capital S, I was using small s. And why? Because when I was writing the sequence, a pop-up showed up in console asking me, are you writing sequence? And I said, yes. I click on it. And then I wrote rest of the command. But I did not make sure that the pop-up opened up while showing the sequence word with a small s in all small letters. And I needed the one with capital S. So I made a mistake and I just accepted that pop-up. And when I click enter, it was showing me error. Then I figured out actually, and then instead of using small cap, I used capital S and click enter, and then click another command, plot as factor sequence. Once you click on that command, hit enter, then this showed up. So, for each command line, it's not necessary that something showed up. Sometimes you're specifying using those command lines to telling the RStudio how to use that command line, right? So for example, when I write sequence PDB seq, this first line, nothing will show up. But also the error should not show up, right? So nothing shows up because here I'm telling the RStudio that the sequence from PDB, fetch it and then show me as factor. But before I have to specify which sequence I'm talking about. So I'm talking about the sequence from the PDB file that is just fetched by the RStudio into the R, right? So here I gave the command. When I hit enter, nothing shows up. So here I'm telling RStudio how to use this command. Now this command, plot as factor sequence, this is what I'm looking for information. When I click enter, and a plot like this will show up in the bottom right pane of the RStudio, where it is supposed to show up graphics and the plots, right? So here it is telling me that the cysteine residue and the tryptophan W is in the lowest quantity or ratio in my protein. The highest ones are alanine, glycine. These are the highest, found in the highest ratio in my protein sequence. So once you are there, and because you are using your own PDB ID, it should show differently for your protein of interest. right? And okay, now you can also do a blast using that protein sequence. And in blast, it will tell you that your protein sequence is similar to how many proteins, right? So the PDB file has been fetched by the R studio into my studio, but it is containing the sequence of only one protein, which is my protein of interest. Now I want to use R to find out, to do blast on it and to find out how many more protein sequences are similar to my protein sequence, the one that I used, that the one which is contained in the PDB file. For this, I will use blast function, and this is a command that you need to write: blast result and sequence, everything that you see in the bold, right? And once you have this, enter, and then after you need to write attributes blast result. So, once the R Studio has done the blast, it won't show you the results unless you ask to please write attributes of it. And these are when you write attribute, this is something showed up in the console. It's mean click on this hit.tbl and try to get the information contained under this command line. And then again, you have to write a command that contains, sorry, I think I said hit, don't hit, but you have to use this as a command line and then try to see what information is contained in this command. Similarly, RAW, you write it down and then see what command it contains in there, right? So when I write attributes blast result, this is what I get. Now I want to see what information is contained in this hit.tbl. I will write another command, head blast result $hit.tbl. If you want to see what is contained in RAW, so instead of hit.tbl, you will write, TBL is for table. So instead of writing hit.tbl, you write RAW and you see what information is contained in there. So when I click on this, it will show me a table, this table here, and it's showing the 5 protein sequences, 1, 2, 3, 4, 5, no, or 6. Actually 6, it's showing me 6 protein sequences from the 6 different proteins and comparing them. What is the name of the protein? What is the identity, alignment, length, mismatches, the I think bit score, and everything that the system compares up with these proteins with each other. You see there are 3 different rows. Actually, there are not 3 different rows because it's difficult to display all these 6 proteins in as one row in a straight line. The system breaks it down into 3 rows. Basically, there are only 6 proteins. The same 6 proteins are shown here, the same are shown here, right? If I stretch this box towards right, you'll be able to see all the 6 proteins are shown in the same rows, right? So now what is the bit score of most similar hit to your protein sequence? In order to make sure students actually are doing this step, I ask them to, once you've done this step, and then look at the bit score. Where is bit score? Here. So for me, my bit score is 1177, right? So I ask them to find out your bit score, the highest one that you find with your protein of interest, right? Next, I want to do one more thing. So, I know that my the R Studio has done the blast search on my protein of interest and has shown me 6 proteins which are similar. I wanna dig down this result further, and for this I will write plot blast result in console so that when I write this command line in console, something like this will show up. This console is having graphs for each of the parameter that the blast search has found out while comparing 6 proteins. The first one will tell you about length. This is the percent identity, it's a bit score, and these are the e-values, right? And what is it telling me? That, it's using a cutoff score of 381. So when the blast result uses a cutoff score of 381, what it finds that out of 6 proteins, these are the ones, they are below the cutoff, the one which are showing as black. So these are the ones we are below the cutoff. If I use a cutoff 381, then out of 381, only 36 hits, they are similar to each other, right? they pass the threshold. So you can specify any cutoff number. If you're looking for example, amino acid sequence that which is similar in all the proteins, and you want to confine that sequence to only 100 amino acid residues, and you say out of 100 amino acid residues, how many are similar, how many proteins have similar sequences, should not be more than 100, under 100. And then if you write down 100 instead of 381, when you write this command, it will give you the 100 will be as the threshold used by the RStudio to give you all the lower values which are less than 100 in black, and all the ones which are on crossing the threshold will be shown as red, right? So you can specify your own threshold value and search what is the best cutoff that you're getting. So here, if you look at the cutoff that they have used, the hits that they are using for different values, which are displayed in this blast result, for log, I'm always getting 36 hit, right? So what actually is telling me, in order to get 36 hits, what is the cutoff used by the system? So in order to get 36 hits, the cutoff used for log E was 381. In order to get 36 hits, the cutoff used for bit score values was 481. In order to get 36 hits, so this is how you read it, read these results, right? The chosen cutoff value was 381 and yielding hits were 36 in number. Similarly, you can change the number of hits. When you change the number of hits, for example, I changed from 381 to 100, I found 37 hits. So I want a student to do this by themselves. And that's all about the R exercise that we learned today. I tried, rather than giving you 5 lectures on explaining what R is, how does it work, what are the functioning, what are the command lines, how to write the command lines, and then 5 lectures about, what are the PDB files and this and that. I tried, and unfortunately, you do not have that much time, and people also learn the theory first and then writing the command line and then be able to write it. But again, in order to learn the applied nature of those command lines, another set of 5 lectures. It was a very tough task and challenging for me to put all this information together in half an hour lecture, where I teach people who have no idea about programming, that what the programming is, which one we are using, how the command lines work, and how you can make use of the information that you are getting through these command lines. And this is the best I come up with. And I hope perhaps some of you have understood some of the stuff. And usually, like by watching simply, you cannot uh, learn better unless you do the experiments by yourself. And this is the reason I designed those questions. So I would highly encourage you, if you are new to the R programming, first try to do it using my sequence, the one that I use, and see if you're getting exactly same results. If you are not, you should be typing your own command. If you're not, copy paste the commands from my slides, and then you should be able to get them. And once you are confident that the system is working for you, then try a different PDB ID, which is your protein, and then do exactly same thing, and then see what information you are getting and what you're not. And the ones you're not getting, why you're not getting that information. Maybe that information is not contained in the exercise, in the PDB file. So with this, I think I conclude today's class. And I had some quizzes that somebody asked me, but I didn't find time to put them in Menti. Maybe in the last lecture, I would ask, we'll have a fun, exercise when we are done the last session, where I ask you these different questions through Menti, and we will all be able to participate. I'm here okay, what I'm doing here now, the second thing is, once you're done, I want to show you the video. And if you just give me 1 second, I want to open the video, and so that you know how we did okay, I have to simply, I think I need to, minimize it. Just give me 1 second. Let me open the video so that you see how the R exercise looks like. I have actually 2 videos, but I'm trying to show you only our video because it's something new for the multiple sequence alignment and the phylogenetic analysis. It was similar to what we have done in the last two exercises. You see the video. So this is so I'm just showing you the first video first, the multiple sequence alignment and the phylogenetic analysis. So while you're watching this, and then I will open the R video after that. Let me know.
In this video, we will be taking a protein sequence, comparing it to other proteins with similar sequences. We will then take the sequence of the 5 most similar proteins and perform a multiple sequence alignment using the cluster omega program. Using the multiple sequence alignment, we will be able to compare which sequences are most similar to each other and construct a phylogenetic tree depicting the relationship between these proteins. First, go to UniProt. Type the ID of your chosen protein in the search bar here. I will be using the sequence for the urease subunit alpha protein Helicobacter pylori. On the left, click on the sequence section in the quick access list here. This will bring you to the section with the sequence. On the right of this section, you can perform a blast run of your sequence directly from UniProt. Hit go, then hit run blast. On this page, you will notice the job identifier in the center. This is associated with our current blast run. If at any time in the future we would like to bring up the same result without running the blast search again, we can do so with this ID. This may take a few seconds or a few minutes, depending on your sequence. Once the run is complete, you will be greeted with the overview section. Scroll down a little bit to find the alignment section. This section contains the alignments list, which shows the most similar proteins based on their sequences. On the right, you will find the E-value, the score, and the percent identity of each of the sequences in relation to your own sequence. Next, we want to download a FASTA file containing the sequences of the top 5 hits. We will use this FASTA file to perform the multiple sequence alignment later on. Select the top 5 hits, like so. Note, do not select your query. Click on the download button. Ensure that you're only downloading the selected 5, that it's FASTA format, that it's uncompressed. Then hit go. Right-click this page and hit Save As. Change the file extension from txt to FASTA and hit Save. Now that you have downloaded the FASTA file with the sequences, we will compare them to each other by performing a multiple sequence alignment. A multiple sequence alignment is an alignment of 3 or more biological protein or nucleic acid sequences. Usually, these sequences are of similar length, and from the output, evolutionary relationships.
Again, the video is not visible and it was also static on one slide. The video is not streaming properly.
Really? Because I am sharing my screen. I have no idea what is happening. 1 second. Because it is on the, what is not, like the video is not working or just static? Or you're not seeing it?
Previously, it was static. Now I cannot see it. It is same as it was before. We cannot see anything.
Not.
And it appeared again right now. And right now it appeared, and I'm not sure how it will go will be static, or I'm not sure.
But right now it's just working, like you can see it's moving. Is it right? Like, the person is typing something in the browser. Do you see that? It's working?
This is the European Bioinformatics
Is
Institute
it working?
Cluster
Like you can
Momentum
see that?
which runs the cluster
Or is it
momentum
static?
algorithm performing multiple sequence alignment. Leave the input type as protein since we are running a protein.
It's working.
Okay, thank you.
Then, open the file that you downloaded earlier, the FASTA file, and paste the entire FASTA file's content into this box here. Scroll down and hit submit. Once the program has completed, you can view the multiple sequence alignment in the Alignments tab, which is already open by default. You can also download the alignment file here, or you can view the phylogenetic tree. Some typical use cases would be to download the alignment file and view it in some sort of multiple sequence alignment viewer. However, we're going to be looking at the phylogenetic tree, so go ahead and click on the phylogenetic tree tab here. Phylogenetic tree is a diagram which shows the evolutionary relationships between species, organisms, or sequences. In this case, a multiple sequence alignment is performed, which compared the sequences to each other and maps out their positions in the tree based on how similar they are to each other. Sequences closer to each other are more similar. Another way to perform the multiple sequence alignment in phylogenetic tree is to use UniProt's built-in multiple sequence alignment tool. Go to UniProt. And at the top, select Align, which is here. One benefit to using UniProt's built-in tool is if you are aligning protein sequences, you can use the UniProt IDs of the proteins rather than the FASTA file. But since we already have the FASTA file sequences, you can just paste them in manually here. Then hit Run Align. Once the program has finished, hit the download button here. Make sure that it's compressed. And hit Go. Open the file and extract the contents. You now have a multiple sequence alignment file denoted by the ALN extension of the 5 sequences you aligned earlier. Keep it here because you will need it for the next step. Go back to the European Bioinformatics Institute website, but this time use their simple phylogeny tool. This is a simple phylogeny tool. You can drag the alignment file into the Choose File section. We're going to upload the multiple sequence alignment file directly. Note that we will be using the neighborhood joining method for clustering, which is sufficient for smaller sets of sequences. If you are running a larger multiple sequence alignment, it would be recommended to use UPGMA as the algorithm instead of neighborhood joining. Hit Submit at the bottom. Once the program is finished, you will find your phylogenetic tree. That concludes this video. You should now know how to compare your protein sequence to another similar protein sequence using UniProt's built-in BLAST program. Then generate a multiple sequence alignment file from the top few hits. Then, using the multiple sequence alignment file, know how to generate a phylogenetic tree to visualize the relationship between your group of proteins. In this video, we will be taking...
So that was all about phylogenetic analysis, and let me show you the another video about R programming in a Bio 3D, and please let me know if it is not visible, because I don't know what happens. I think sometimes the screen stops sharing, and I don't know unless somebody let me know in the chat, or just please unmute yourself.
In this video, we will be using R to briefly analyze a protein using the Bio 3D package. We will be looking at the attributes of a protein, its sequence, and performing a BLAST search in R of our protein. First, make sure that you have both R version 4.1.1 installed and RStudio. R is the program that runs the code, while RStudio is a graphical interface that makes it easier as a user to interact with R. We need to first install the Bio 3D package. A package is an extension to the language that contains code, data, and documentation, and are free to use. From the menu, select Tools. Then, select Install Packages. Under Packages, type in Bio 3D and hit Install. Now that we have it installed, we must load the package. We can load it with the following command: library, and in brackets, Bio3D, the package we just installed. It's now loaded. Next, we'll want to assign a protein structure to a variable. We'll call our variable PDB underscore ID, and we'll use the arrow to denote that we're assigning the results of whatever is on the right to our variable. We'll be using 1E9Z, which is the crystal structure of Helicobacter pylori urease. Hit enter. Next, we will use the read.pdb function to access the PDB database and find the protein structure with ID 1E9Z, then assign it to a new variable called PDB. First, I'll name the variable PDB, and using the arrow, assign the results of read.pdb and then our PDB ID. We can now print the contents of PDB using print and then what we're printing in brackets. Hit enter. You'll notice that you can find the sequence, attributes, and other information of the protein in PDB. Let's say we wanted to view the attributes of our protein. We could use attributes PDB, like so, and hit enter. If we wanted to only view, let's say, the atom attributes, we could use PDB $atom. To view all the atoms in the structure, the $sign is used to access an individual attribute. If ever you want to see what functions you can perform with Bio3D or any other package, you can type ? and then the name of the package for more information. You'll find information on the package in the bottom right pane of RStudio under the Help tab. Let's look at the sequence of our protein. Type PDB-seq for sequence, and then our PDB variable that we saved earlier. You'll see the amino acid and its position laid out in tabular form. We can also save this variable. Let's call this variable sequence and save the contents of PDB-seq, PDB, the function we just ran, to sequence. That way, if we want to view the contents of sequence, we don't have to run the function again, and we can just type sequence to view its contents. Additionally, R has many visualization tools. We can visualize the sequence object we just saved to view the frequencies of amino acids in a histogram. Type plot as.factor and in brackets sequence, which is the variable we just saved. Note, if you didn't save the contents of PDB-seq in sequence, you could also do PDB-seq brackets PDB, which is the same thing. On the bottom right pane of RStudio, under the plots, you'll notice a histogram after running the command. Here we can see the guanines being the most common amino acid in our protein. In R, we can also use the Bio3D package to perform BLAST searches. Let us make a new variable called BLAST result. And save the results of blast.pdb and then the sequence. This will run a blast search on the sequence of our protein. This may take a few seconds. We can view the attributes of the blast search by using the attributes function, like so. Here we are viewing the attributes of our blast result. Notice that there are three attributes: hit.table, raw, and URL. Hit.table is a data frame summarizing blast results for each reported hit. Raw is the raw blast output, and URL is the single element character vector with the NCBI result URL and RID code. Let's look into hit.table a little more. Type head blast result, the money sign, and hit.table. See the first few hits in the table. Head is a function that shows the first few lines in an entry, such as a table, list, or an array. In this case, using head on hit.table brings up the first 6 hits from the table as a result of blasting our PDB query. Since these hits are already sorted, the first row should be the most similar protein to our query. You will see the E value and the bit score here as well. Note that sometimes the results are broken up over 2 lines. Lastly, you can also plot the blast results by using plot blast result. On the bottom right pane, once again, under the Plots tab, you'll see the blast results plotted with E-value, bit score, identity, and length. The black hits have passed the cutoff threshold, and the red ones were unable to pass the threshold. In this case, for the E-value, the cutoff threshold was X of 381. In general, the higher the cutoff value, the smaller number of hits we get. We can also set the cutoff value, like so. I will arbitrarily set the cutoff to 100. Notice, if we compare the plots from before with a cutoff value of 381, we had 36 hits. And when I chose a cutoff value of 100, I got 37 hits. That concludes this video. You should now know how to install the Bio3D package and run basic protein structure analysis using the package. You should also know how to view the sequence contents. And you should be able to run a blast search on a protein sequence and plot the blast results using the rplot function.
Okay, I think with this, we are done today's exercise. So let me see if you have any questions. You're most welcome to write in the chat or unmute yourself and speak, ask a question on the mic. Any question about, like the exercises, if you have tried to doing it, and about today's exercise. Let me see if there is any question in the chat. Slides. So Muzammil, I think the organizers, they will share with you the video links because all these sessions are being recorded, and we have not talked about sharing the slides. Perhaps we can see the possibility. If it is possible, yes, I will be happy to share the slides with the participants. But only with those participants who are here in the class, who attended all the sessions, were there till the end. Those are the ones actually will be getting the slides. And I think you'll be getting a micro credential, but again, the requirement is all the students, or I think a digital badge you'll be getting. The requirement is you are supposed to attend all the sessions and will be here till the end. Because then it just makes sense then, if you're showing that you have done something, that you should be able to reproduce the results and the data and the information, the knowledge that you gain through the workshop or through the course. Okay, somebody wrote me a very good answer there when I was asking that why plants hate math is plant leaves are difficult to count. Yeah, I think I showed the right answer. So sorry, I couldn't see that answer when you wrote it at that time. Maybe it was late or what. And yeah. And I think I already mentioned how to use Blast in the today's exercise, but in the last two also, I think I mentioned how to use Blast. Okay, I don't see any more questions in the chat. And with this, I think we conclude today's session. Have a good night. The ones who are having a night, have a good day for the people who are joining in from the EST time zone. And I'll see you on Friday with the 4th session. Have a good day. Bye-bye.